Hi, I'm Dave Moran. I'm the co-author of the Visual Histology DVD series. This film clip is to give you a brief introduction to what one of the individual DVDs is like.
This image is an electron micrograph of a pancreatic acinar cell, which is a cell which is involved with very rapid rates of protein synthesis and secretion. This image comes from figure 1-2 in your visual histology textbook, and the arrow right now is in the nucleus. The nucleolus is here, the rough endoplasmic reticulum is here, secretory vesicles are here, this is a mitochondrion right here, and in particular I want to call your attention to the cell membrane out here. Now this little thin line is the cell membrane, which is the outer limit of the cell, and it's shown at higher magnification here, and what we see here really are two cell membranes, one from this cell here and one from the cell above it, that are next to each other, and between them is the so-called intercellular space. Now in the next image we'll look at this at higher magnification. Here the electron microscope shows us two cell membranes next to each other with the intervening intercellular space. The arrow is pointing at the cell membrane of the lower cell. Now it points at the cell membrane of the upper cell, and now the point of the arrow is in the intercellular space. And if you look carefully, you notice that each cell membrane looks like a pair of railroad tracks. There are two dark outer lines and a white clear inner line. This is the characteristic appearance of the cell membrane under the electron microscope. When first seen, this was called the unit membrane because membranes that surround cells look ultrastructurally like the membranes that surround cellular organelles. Biochemically, they can be very different. There's a major take-home point that I want you to think about, and that is that although the cell membrane looks quite similar along its entire circumference, patches of cell membrane can vary dramatically from one area to another. So the homogeneous structure you see under the electron microscope does not give a clue as to the tremendous biochemical diversity manifest by cell membranes at different points along their borders. This is a light microscopic image of the same cells we were just looking at electron microscopically. This is a group of pancreatic exocrine cells. They're also called pancreatic acinar cells, and they're so called because a ball of these cells is organized into a secretory unit called an acinus. In this case, you have a lot of cells like this one here, this one here, this one here, that are all forming these secretory granules that are pouring into the lumen at the center of the acinus. Now this preparation is a light micrograph of a preparation that was prepared as you would prepare specimens for electron microscopy, so a tremendous amount of detail can be seen even at the level of the light microscope. This is not like standard histological preparations, but let's just have a look here. You have the nucleus here, nucleolus here, and these dark streaks are the mitochondria. You can actually see these quite clearly in this preparation. This dark area here is the rough endoplasmic reticulum. These dark little BBs are the secretory granules, sometimes called secretory vesicles, sometimes called secretory droplets, and this is the lumen into which they all pour. Now this is a light microscopic image of a cell that's very different from the pancreatic acinar cell, but still has some of the same components. This is a large motor neuron from the spinal cord, and we're looking at the cell body here, and I'll trace the approximate contours of the cell body with the arrow here. Now the motor neuron has a large centrally located nucleus here, and it's mostly clear. It's mostly euchromatic. There's a dense nucleolus in the middle, and then you can see some sort of purplish patches. Now this is stained with toluidine blue. In toluidine blue sections, the rough endoplasmic reticulum stains either blue or purple, and these clumps here are clumps of rough endoplasmic reticulum and free ribosomes. In nerves they're called the Nissl substance, and out here you can see little piles of dark staining sort of BBs. These are lysosomes that have ingested something from the outside world and have been unable to completely degrade the materials. So these structures are called lipofuscin granules. They're also called wear and tear granules, and they represent lysosomes that have glommed onto something that they cannot digest. In nerve cells, lipofuscin granules or wear and tear granules tend to build up with age. So the more you see in general, the older the nerve cell. Lysosomes tend to be too small to see with the light microscope, but when they've combined with something indigestible and formed these large complexes. There's another kind of very important cell surface specialization called the microvillus, and the plural is microvilli. This is a light micrograph of an intestinal villus, and each of the intestinal epithelial cells here is covered by a group of microvilli. Now, they're too small to individually resolve with the light microscope, but taken together they form this structure here, which is called the brush border, which is a whole bunch of microvilli arranged in a row. Now let's have a look at this at higher magnification on the next slide. This is a light microscopic image of the same specimen we were looking at, but taken at higher magnification.